Okay, we're going to talk about the history of the discovery of DNA and how our ideas have developed with it. Right, in 1929, it was known that DNA was made up of uh, deoxyribose sugar, phosphate groups, and nitrogenous bases, adenine, thymidine, guanine, and cytosine. But it wasn't known how they were joined together. And also, they knew that DNA was in the nucleus, but most people didn't think that it carried the genetic material. At that point, people believed that protein was probably the molecule which carried the genetic material, because protein appeared to be much more complicated. DNA was too simple a molecule, but they couldn't really see how it could encode information. Right. Right. Now, the first bit of evidence that suggested that DNA was the genetic material comes 1943, a group led by Avery. He was investigating the pneumococcus bug. Right, it's a bacteria which causes pneumonia. Now, bacteria can undergo sexual reproduction. Now, what is sexual reproduction? Sexual reproduction is when you get two organisms which exchange genetic material. And bacteria, generally speaking, we think of them doing as asexual reproduction, but they can exchange genetic information. In 1943, no one knew what the genetic information was. They thought it was probably protein. Right, so the two kinds of pneumococcus, there's a blue one there, which was called the smooth type, and that was lethal. So if a mouse is injected that, they develop pneumonia and die. And then the green type, it's called the rough, and that was harmless. So the mouse was injected with that, nothing would happen to it. Right, so Avery, they did an experiment. They got the rough kind of bacteria, the harmless one, and they added some the dead smooth type. Right, so that was dead, it wasn't alive. And they found out that what happened was the rough ones, sometimes they turned into this new smooth version, which was lethal. And they said they had been transformed. And they call this process transformation. It's really a kind of sexual reproduction. And they wanted to know what was the substance passing from the smooth here into the rough that was causing this change in phenotype, in appearance. So they set about adding proteinases to they got the dead smooth type. They added proteinases, digest all the proteins. And they found out that didn't do anything. The extract was still perfectly capable of transforming the bacteria, suggested the genetic material was not protein. They did the same with carbohydrates and so forth, and eventually they narrowed it down to DNA. They actually extracted pure DNA from the dead smooth ones, and they found it was like a filamentous white molecule, which you may have done yourself if you can extract DNA from like kiwi fruit or onions and stuff in the school lab. And when they added this filamentous material to the rough, they could turn it into the lethal type. So they concluded from this that DNA was the genetic material. Right, now the next big thing here was the Chargaff Rules. Now Chargaff, he didn't know anything about the double helix, which I've drawn a picture of here. And I'm sort of going backwards here, but what you can see from the double helix, because each strand pairs up with the other strand, and you know that T always pairs with A and G always pairs with C. Well, because of that, it would follow, if that was the case, that the percentage of, if you look at all the DNA in an organism, the percentage G has always got to equal to the percentage of C, and the percentage of T has always got to equal to the percentage of A. Now, Chargaff found out that this was true. This is his first rule, this one here. And later on, he didn't know anything about the double helix, but of course, Watson and Crick, later on, they knew about Chargaff's work, and that helped them to, predict that you're going to get this pairing here. So Chargaff found that out. I think in human beings, it's something like you get about 20% G and 20% C and 30% T and 30% A. He also did it. He did it for about 5 or 6 different species. I think in yeast it's slightly higher. It's like in yeast he found out it was 33% T and 33% A, and 17 of C and 17 of G. Now that leads me on to the second of Chargaff's rules, which he said different percentages of GC and TA for different species. And that tends to suggest that maybe the amount of those things is something to do with the genetic code. Was it interesting? If you looked at the protein, the different amino acids in human beings and in yeast or whatever, you find out that the percentage of amino acids are actually pretty similar. So that was another bit of evidence that it was nucleic acid, DNA, and not protein that was the genetic material. So they were Chargaff's rules in 1949. Then let's go on to the most famous, 1953. Watson and Crick produced their paper, which suggested the double helix. Where do they get their evidence from? Well, from lots of different places, things like Chargaff's ideas, but one of the really important things was X-ray crystallography. Okay, now X-ray crystallography, you can't see the molecule with an X-ray at all. You can't see that, but the X-rays will diffract. If you know physics GCSE, what diffraction is, the X-rays will be diffracted by crystals, and you can detect the X-rays on a photographic plate, and you get a pattern of different spots and bands and things. From then, you can work out. Well, they will manage to work out. They got certain clues about the structure of DNA from that. That didn't tell them the entire structure. And then they use a lot of other information on things like bond lengths. They knew the lengths of bonds between things, and they knew the bond angles and so forth. And they built up a model, and the model fitted all the X-ray data and all the other information about bond lengths and so on. There was a little bit of controversy here because the X-ray pictures were developed by a woman called Rosalind Franklin, and she died fairly shortly after she made these pictures. And really, Watson and Crick didn't give her the full credit for these pictures, and some of the pictures they managed to see without her permission. Her boss showed them to Watson and Crick without telling her, and there's a lot of controversy there. She didn't get a Nobel Prize. Some people suggest that she should have shared the Nobel Prize with Watson and Crick and Franklin's boss. Right. Anyway, let's get on to the next thing, the Meselson style experiment, 1958. Right, what was that about? Right. Well, this proved something called that DNA was replicated. Sorry. The replication of DNA was semi-conservative. And I'll explain what that means here. So semi-conservative replication and Watson and Crick hinted, they didn't call it that, but they hinted in their original paper that that's probably how DNA did replicate. Because, right, what they said was, right, you've got, there's a strand of DNA with and you've got your bases all along it, okay, your sequence of bases. What happens when it gets replicated? One of those red strands, that acts as a template. Okay, so it splits into 2 strands, yeah? Each strand acts as a template, and you make the new strand, which I've colored in blue there, and they use the red strand as a template. That would be semi-conservative replication, and everyone hoped that what would happen, because it would mean that the order of the bases would be conserved, and that would mean that the order of bases probably did encode the genetic material, and that would lead a way forward to understand the genetic code. Right, I suppose the other way in which replication, if it was conservative replication, this was another possibility, where you got one strand of DNA, you got your double strand of DNA, you kept that intact, and then you produced a brand new strand, and both strands were brand new. And this was less appealing, this one really, because it didn't really explain how the order of the bases could be conserved. So everyone was hoping, yeah, semi-conservative replication, that was the most likely thing, and that would lead to a lot more discoveries. Now, Meselson Stahl did the experiment, which managed to show this in 1958. Right, I've just drawn a little picture here of the base pairs. Yeah, these are the bases, GC and AT, and you'll see in those you've got an awful lot of nitrogen atoms. Now, nitrogen normally comes in the isotope 14N. There's a tiny proportion of it, which is this heavy one, 15N. That is not, 15N is not radioactive. It's just merely, it's stable. It's just heavier than the 14N. So if you made DNA with bases, which are all 14N, and the DNA was all 15N, sorry, the DNA would be heavier than if it was made of 14, and it would be lighter. So that's how they, that was the way in which they proposed to tell the difference between 15N strands and 14N. So let's see what they did. So, right, I'm going to get rid of that. And that. So here's Meselson Stahl experiment. They got a bacteria, E. coli, which is very easy to grow. It's found in your gut. It's very easy to grow, and that's why they chose it. And you can grow it in medium, so it's growing here. You've got culture medium, you just give it all the things it needs, like glucose and stuff. But what they did was they grew it in medium which contained nucleotides, so they're the building blocks for a DNA. So, like, you know, you've got your nucleotide with G, one with C, and one with T, and then A. So they added them to the medium, but these were not with normal 14N, with 15N. So these E. coli ended up with a load of DNA which would be heavier than you'd normally get. Right, so I'm going to draw that double-stranded DNA here. We've got, and I'm going to color code those. Let's have the 15 as red, and we'll do the 14N as blue. So what they did then was they got those E. coli and they extracted them from the culture medium by putting them in the centrifuge. They all go down to the bottom of the tube, throw away the culture medium, and they put them in normal 14N medium there. So I should draw that in blue. I'll do that in blue to get right. And so they put them in there, and then they allowed them to divide once. They got the bacteria, and they extracted the DNA from those bacteria. So what's going to happen there is if you get semi-conservative replication, you would end up with DNA which contains one strand of 15 and one of 14. So this DNA wouldn't be heavy, or this would be intermediate, wouldn't it? This would be an intermediate heaviness. So they allowed that to happen. And then they allowed the cells to divide once more. So they left them in a little bit longer that time. And they said, right, and they removed and extracted the DNA. And they said, right, if you get the DNA that time, then let's see what happens. Let's do these two strands separately. So that strand, right, you're going to get the red one acting as a template, and then you get the new DNA, which is blue, the fifth 14. So that'd be a mixture of the two, a medium density. And then the blue strand would act as a template, and you get a new blue strand, and that would give you a this would be the light one, because it would be all 14N, this double blue one here. Likewise here, from this division, that blue strand has a template, and the new strand it makes is with 14, so that's blue. And then this one, it acts as the red strand is the template, and you build up the new strand, which is 14. Okay, so you can see this one and this one would be the medium density, and these two would be light. So how do they get the DNA and find out where it was heavy or light? Well, they used something called a density gradient, which I'm trying to show here. Okay, density gradient. So you get a tube here, and you make a solution of cesium chloride in that tube. Now, cesium ions are very heavy, and when you put this in a centrifuge and you spin it, what happens? All the heavy things will go to the bottom. Now, because cesium is so heavy, it tends to, you get a more concentrated solution at the bottom, so you can see that's happening in gradient, yeah? It gets more concentrated as you go down the tube. You have to spin it for a long time, like 24 hours or something. And at the top end, it will be less cesium, more dilute. Now, the more cesium you've got dissolved in there, the more dense it is. Okay, so, and the DNA will float at its own natural buoyancy. So say you get a piece of DNA, I'll do it in purple, and you might get it floating there. Okay, so that means the DNA has got the same buoyancy, the same density as the cesium chloride at that point. Or if the DNA was a bit heavier, then it would end up floating a bit further down the tube. Or if it was really heavy, then it would end up near towards the bottom there. Okay, so the DNA will tend to sort of float out at its natural level. So once you've spun your DNA in the density gradient, it's very easy to see the DNA. You can see it. All you do is you look at it under UV light, and it absorbs the UV light, so you can see a dark band in the test tube. Right, let's see what happened when they did this experiment. So here's the first density gradient they did. So I'll just make a bit of space here. Okay, so for this one, I'll draw a tube here. Right, you've only got this heavy stuff, the 15N, so that's going to go for quite far down the tube, so I'll do that there. I'll do it in red. So see a band of DNA with the E. coli that you haven't allowed to divide, you get just the DNA there. Now, once they'd done this this allowed it to divide once, they did, right, you put this one in there, you would not see the heavy band, the 15, which I'm going to draw in, you don't see it. I'll get rid of that. What you do is you're going to get one which is a bit lighter. And I'll draw that in purple. Okay, so this is the mixture. So you get that band there. And just to show you where the heavy band was, right, the heavy band 15 one would be just there. And if you just had 14, that would be less dense, so it would be up there. So after one division, you don't get any pure 14, you don't get any pure 15, you get this mixture, which is due to the fact you've got one strand in the DNA, one strand is the 15, one strand is the 14. You allow it to divide a second time, and you do your density gradient, you get your tubes there. Right, now in this one, you won't get a pure 15 band, but you will get the mixture, which I'll draw in purple. But you will also get the blue one, which is less dense, which will be higher up the tube. And this proves that you've got semi-conservative replication. And you can see if you allow them to divide some more, all that would happen is you'd never get the pure 15 band back again, but the mixture one would get a bit weaker every time. So I'm trying to show that's shown a bit weaker, and the more and more of the DNA would be the light pure 14 type. So this showed that replication was indeed semi-conservative. And this helped, that showed that one strand of DNA acts as a template for the replication, which would mean that the base sequence is conserved. And if the base sequence was the way the genetic code was encoded, then you would have to have semi-conservative replication. You did get semi-conservative replication, so that was all the more evidence that DNA was the genetic material, which everyone at that point recognized it was.